Today we're doing something a little different. We usually talk about concepts, right? Psychology, history, trends.
The big picture stuff.
Yeah, today we're opening up the hood. We are looking at the engine. We've got a stack of research here based on Chapter 46, Organization of the Nervous System, Basic Functions of Synapses and Neurotransmitters. And we're going to try to decode the actual hardware that makes you possible.
And this is one of my absolute favorite topics because it really bridges that gap between, say, physics and philosophy. We talk about thinking or remembering as if they're these magical spells.
Like they're abstract concepts.
Exactly, but they're not. They are mechanical processes, they're ions, they're electricity, and they are chemistry.
That's the mission today. I want to understand how a physical signal, like a photon, a literal particle of light hitting my retina, eventually becomes the nostalgia I feel when I see a picture of my childhood home.
There's a chain of events there.
A mechanical chain. And we are going to trace it line by line.
From the macro level all the way down to the micro. We'll look at the neurons, the synapses, and the chemicals themselves. Basically, the hardware, the software, and the code.
Let's start with the brick and mortar then. The neuron. You mentioned 100 billion of them in the central nervous system alone, which is a number that's hard to even wrap your head around.
It's just a big number, yeah.
So what actually is a neuron? Because I think most people, myself included, kind of picture a blob with some wires sticking out of it.
Well, that's not a terrible starting point, actually. But the architecture is incredibly specific, and it's designed for a single purpose, processing information. While they come in a lot of shapes and sizes, let's call the typical design has three very distinct parts that define its function.
Okay.
You have the soma, the axon, and the dendrites.
All right, let's break those down. The soma.
The soma is the body. Think of it as the main chassis of the cell. It's where the nucleus lives. It's where all the metabolic machinery is humming away, keeping the lights on. It's really the headquarters for that individual cell.
So it's the command center. And the axon.
The axon is the output cable. It's this single long projection that extends out from the soma, sometimes for a very long distance. Its whole job is to carry the signal away from the neuron to somewhere else.
So the axon is the mouth of the neuron. It's speaking to its neighbors.
Perfectly put. And then you have the dendrites. These are the ears.
Okay.
These are these huge numbers of branching projections that stick out from the soma. They can spread out for, up to 1 millimeter into the surrounding tissue. This is the main input zone.
And this is where that 200,000 connections stat comes in, right? This is where it's listening.
It is a single neuron on its surface, mostly on its dendrites, but also on the soma. It's covered in these synaptic connections, these little docking ports. And they can number from just a few hundred to, yes, as many as 200,000.
Let's just pause on that for a second. If I'm a single neuron, I am just sitting there, and I could have 200,000 different people shouting instructions at me at the exact same time.
Yes. And it's total chaos. Some are shouting fire, some are shouting don't fire, go left, stop, more, less. It's this constant chaotic roar of information.
So how does a neuron not just have a nervous breakdown?
Well, that right there, that is the essence of what the text calls the integrative function of the nervous system. The neuron's one job is to take that roar, do the math, and it's literal math, summing up positive and negative charges, and then decide whether or not to send one single clean signal down its axon to the next guy.
So it turns all of that chaos into a simple binary decision, a yes or a no.
Precisely. And for that to work, for the whole system not to descend into madness, there is a fundamental traffic law in the nervous system. The source material calls it the one-way rule.
Then what's that?
In the overwhelming majority of synapses, what we call chemical synapses, the signal always travels from the axon of the preceding neuron to the dendrites or soma of the next neuron. It never, ever goes backward.
Why is that so critical? I mean, a wire in my house can carry electricity both ways if I hooked it up wrong.
Imagine if your nerves worked both ways. You touch a hot stove, the signal zips up to your brain saying hot, but then it bounces right back down the same wire, hits your finger again, bounces back up to the brain.
It would be an infinite feedback loop.
An infinite loop. You'd have a seizure. You'd have absolute chaos. The one-way rule ensures that a sensation leads to processing, and that processing leads to a specific action. It gives the whole system a purpose, a goal.
Okay, so we've got the building blocks,
Yeah.
the neuron. Now let's zoom out and look at the layout of the city. The source material divides the whole nervous system into two massive buckets: sensory and motor, input and output.
Correct. The sensory division is the Let's call it the experience department.
And this all starts at the edges of the body, right? The periphery.
Right. You've got these specialized receptors all over your body surface and inside you, visual in your eyes, auditory in your ears, tactile in your skin. So let's say you touch a rough piece of sandpaper.
Okay.
That tactile experience excites a sensory receptor in the tip of your finger.
And that one little signal starts a journey.
A huge journey. It travels up through the peripheral nerves into the spinal cord, and from there it branches out. It hits multiple levels almost simultaneously, the reticular substance in the medulla, the cerebellum, the thalamus, and eventually it reaches the penthouse suite, the cerebral cortex.
It's like a ladder, but it's hitting multiple rungs at the same time.
It is. But remember, this is all just the flow of information. And information is totally useless unless you do something with it. And that brings us to the motor division.
The effectors. I love that term. It sounds very sci-fi.
It's proactive, isn't it?
Yeah.
The eventual ultimate role of the nervous system is to control bodily activities. And when you boil it all down, the brain only has 3 ways to interact with the physical universe.
Only 3. That seems low.
Only 3. It can contract a skeletal muscle like moving your arm to pick up a coffee cup or using your vocal cords to speak.
Okay, 1.
It can contract smooth muscle, that's things like churning your stomach or constricting a blood vessel to raise your blood pressure, or it can cause the secretion of active chemical substances from glands, adrenaline, hormones, saliva.
That's it. Everything I've ever done in my entire life. Building a house, writing a poem, hugging my kids. It's just a combination of muscle contractions and squirting chemicals.
At the output level, yes. That is your entire interface with reality. And running in parallel to this, of course, is the autonomic nervous system, which handles all the internal housekeeping, heart rate, digestion, mostly subconsciously. But the mechanism is identical, contraction and secretion.
But there's this huge space between the input, the sandpaper, and the output, moving my hand. And this is that magic middle ground. The text calls it the integrative function, and it uses an analogy that I think is more relevant today than ever, the spam filter.
This might be the single most important concept to grasp about your own brain. Your brain is not a recording device. It's not a camera. It acts more like an editor.
A really, really ruthless editor.
Extremely ruthless. The statistic in the source material is just staggering. More than 99% of all sensory information is discarded by the brain as irrelevant and unimportant.
99%. So I'm basically missing almost everything that's happening around me.
You have to. You absolutely have to. Think about it right now. Can you feel the pressure of your socks on your ankles?
I mean,
Yep.
I can now that you've said it.
Exactly. But 10 seconds ago, you couldn't. Your tactile receptors were screaming, socks, 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 sending that signal all the way up to your brain, but your brain was just blocking it. It was deemed spam, totally irrelevant to your survival or your current task.
So the contact with the seat I'm sitting on, the hum of the lights in this room.
All of it, the slight breeze from the vent, the peripheral visual noise that isn't moving, it's all being actively suppressed.
Because if it wasn't,
you'd be paralyzed. You would be so overwhelmed by data you couldn't function. The integrative function is the brain's remarkable ability to select only the most important, the most actionable information, channel it to the proper integrative and motor regions, and then generate an appropriate response. It's not just about receiving, it's about deciding what matters.
so we have this editor, this spam filter.
Yeah.
But the editor lives on different floors of the building, so to speak. The source material breaks down the nervous system into 3 specific functional levels. And I have to admit, this section corrected a major misconception I've had my whole life.
Yeah. Which one?
I always thought the brain was the CEO, the absolute boss, and the spinal cord was just the Ethernet cable that connected the CEO to the workers on the factory floor.
A very common misconception.
But the text says the spinal cord is actually a manager in its own right. This is what it calls level 1, the spinal cord level.
Yes, we tend to think of the spine as just a conduit, a simple wire. But let's do a bit of a gruesome thought experiment. If we were to say, surgically cut the connection between the brain and the spinal cord, high up in the neck region, what can the cord still do on its own?
Well, the assumption would be nothing. Total paralysis from the neck down.
You lose conscious control, that's true, but the neuronal circuits that are contained entirely within the cord are surprisingly sophisticated. For instance, even without any input from the brain, the spinal cord can organize walking movements.
Walking. How is that possible without a brain?
Well, if you support an animal or a person upright, so gravity isn't an issue, the spinal cord can execute the basic pattern. Flex the left leg, extend the right, then flex the right, extend the left. That oscillating circuit is hardwired into the cord itself. It's what we call a central pattern generator.
So the brain isn't micromanaging every single step I take when I go for a walk.
Not even close. The brain basically sends a high-level command that says walk forward, speed 5. The spinal cord receives that and then executes the incredibly complex subroutine of contract quad, relax hamstring, adjust balance, push off with toe.
Wow. So the CEO just says, increase sales. And the regional manager is the one who actually figures out the shift roster and the marketing plan.
That's a perfect analogy. The cord also handles all your withdrawal reflexes, pulling your hand away from a hot pan before your brain even registers the pain. It can stiffen your legs to support your body against gravity. It controls local blood vessels, gastrointestinal movements. The spinal cord is an incredibly sophisticated computer in its own right.
Okay, so let's go up the elevator, level 2, the lower brain, or the subcortical level. Here we're talking about structures like the medulla, the pons, the hypothalamus, the cerebellum.
This is the subconscious powerhouse. This is the life support system for the entire body. It controls things like arterial pressure and respiration. You don't think about keeping your blood pressure at 120/80. The lower brain just does that. It also handles equilibrium, balance.
But the notes here mention something that I found really surprising: emotion. I tend to think of my feelings as these very high-level human traits. You know, I am sad because of the existential weight of the universe.
We do like to flatter ourselves, don't we? But the raw, primal machinery of emotion, anger, excitement, sexual response, the fundamental reaction to pain and pleasure, this all originates here in the lower brain.
Really?
Yes. In experiments where the cortex, the higher brain, is destroyed or removed, animals can still show clear signs of rage, of fear, of sexual drive. The animal drives are deep, they're ancient, and they live in these subcortical structures.
So what does that leave for the penthouse? Level 3, the higher brain, the cortical level, the big wrinkly gray matter we all see in the movies.
The cerebral cortex. This is the great memory storehouse. The cortex is vast, and its primary job is to store the files of our life, our knowledge, our experiences. But here is the critical nuance that the text really emphasizes. The cortex never functions alone.
It's dependent on the lower levels.
Desperately. The cortex is like a massive library or a giant hard drive filled with data and programs and memories. But a hard drive can't turn itself on.
It needs a power supply.
Exactly. The lower brain is the power button. The notes describe it beautifully. The lower brain initiates wakefulness in the cortex. Without those lower brain centers sending constant wake-up signals, these excitatory drives, the cortex is useless. It is just a silent, dark library. It can't access any of its own information.
So the interplay is the lower brain provides the raw drive and the energy, I'm hungry, I'm angry, and the cortex provides the direction and the strategy.
That's it. The cortex opens the world of memory and association to the lower brain's primitive drives. So the lower brain says, I'm hungry. The cortex says, I remember there's a refrigerator in the kitchen, and to open it, we need to perform this complex motor task of pulling the handle. It provides the precision.
It connects the data to the desire.
Precisely. You know, there's a diagram in the sources, I think it's figure sum 6-4, that compares the whole nervous system to a general-purpose computer.
And it feels like a pretty valid comparison.
It is, to a point. You have your input circuits, your output circuits, and a central processing unit.
Right. Senses are the input, muscles are the output.
But the key difference is complexity. In a simple computer or a simple nervous system, the output is controlled directly by the input. It's just reflexes. Input A always leads to output B. But as computers and brains get more complex, the output isn't just determined by the immediate input anymore. It's determined by memory, by all the previous inputs.
That's what makes us us, isn't it? I don't just react to a spider because it's a spider. I react because I have this stored memory of a scary movie I saw when I was 5. Or maybe a bite I got.
Correct. The processing unit acts on its history, not just the present moment. And that is a far more sophisticated level of computation.
All right, we've looked at the city. We've looked at the floors of the building. Now I want to zoom way in. I want to look at the door handles, the synapses. Because this is where the actual communication, the transfer of information happens.
The synapse, the junction point. This is, as you said, where the magic happens.
And the text mentions 2 main types, chemical and electrical.
And it's a really important distinction to make. In the human central nervous system, the vast, vast majority are chemical synapses. This means the first neuron secretes a chemical substance, a neurotransmitter, that has to physically cross a gap to bind to the second neuron.
Okay, so it's sending a messenger. What about the electrical ones?
Those are called gap junctions. They're much simpler. They are literal open fluid channels that directly connect the cytoplasm of two cells. Ions just flow straight through from one cell to the next, like water through a pipe.
That sounds way faster and more efficient. Why don't we just use that for everything?
It is faster, and it's often bidirectional. The signal can go both ways, but it's very hard to control. It's like having a city where every building is connected by open hallways. A signal will just spread everywhere instantly, uncontrollably.
Total chaos again.
Right. Chemical synapses, because they rely on a specific chemical key fitting into a specific lock, they act like a check valve. They enforce that one-way conduction we talked about. This allows the brain to direct signals toward very specific goals. Electrical synapses are great for things that need to fire all at once in perfect synchrony, like a group of muscle fibers in the heart that all need to pump together.
But for complex thinking, for processing, you need the control of chemistry.
You need the control.
So let's visualize this chemical synapse. We're looking at what the text calls the presynaptic terminal. It mentions they look like little knobs or boutons.
Yeah, think of them as these tiny, slightly swollen bulbs at the very end of the axon's branches. A single target neuron might have thousands of these little knobs, studding its dendrites and soma from many different incoming neurons.
If we could shrink down, magic school bus style, and stand inside one of these knobs, what would we see?
You'd be standing in a very busy little factory, and two internal structures would immediately stand out. First, you'd see mitochondria everywhere.
The powerhouse of the cell.
Yeah.
I do remember that from ninth grade biology.
It's a cliche because it's absolutely true. They are working constantly to provide ATP, the energy currency, which is needed to synthesize the neurotransmitter. The terminal has to make its own ammunition right on site.
And that ammunition is stored in?
The synaptic vesicles. These are the storage containers. They are these tiny, membrane-enclosed capsules, and each one is holding a payload of the chemical neurotransmitter. Imagine thousands of little water balloons filled with this chemical, all waiting near the edge of a cliff.
And that cliff is the synaptic cleft.
It's a tiny, tiny chasm, only about 200 to 300 angstroms wide. It separates the presynaptic terminal from the membrane of the next neuron, and the signal has to somehow jump that gap.
Okay, so how does it jump? This is part 4 of our outline. The mechanism of release. This is the moment of truth.
It all comes down to one thing: calcium.
Calcium.
Like from my milk.
The very same ion. But in this context, it acts as the trigger for the gun. When the electrical signal, the action potential, comes rushing down the axon and hits that terminal knob, it causes a rapid change in the voltage of the membrane.
It depolarizes it.
Exactly. And that voltage change flings open some very specific doors in the membrane. The voltage-gated calcium channels.
So the electricity opens the door for the calcium. It opens the door. Now, there is a massive concentration of calcium ions in the fluid outside the cell and very, very little calcium inside. So when those channels open, what do you think happens?
Calcium rushes in.
Rushes in like a flood. And this is the cool part. The calcium ions bind to special protein spots on the inside of the membrane called release sites. It's like the calcium physically grabs those synaptic vesicles, or little water balloons, and forces them to fuse with the cell wall.
And they pop.
They pop open to the outside. The process is called exocytosis. The vesicle opens up and dumps its entire chemical contents into the synaptic cleft. And the quantity of transmitter that gets released is directly proportional to the number of calcium ions that entered. More calcium, bigger signal.
So the chemical is dumped into the gap, it floats across, and then it hits the next neuron, the postsynaptic neuron. What happens there?
It finds its target, a receptor protein. This protein is embedded in the postsynaptic membrane. It has a binding component that sticks out into the cleft to catch the chemical, sort of like a catcher's mitt, perfectly shaped for one type of ball.
And once it catches it,
Once the neurotransmitter binds to that receptor, it triggers a change inside the new neuron. And this is where the road forks. There are two main ways a receptor can react, and we really need to understand this distinction because it basically explains the difference between a simple reflex and a long-term memory.
okay, break it down for me.
The first way is the fast and direct route. It's called an ionotropic receptor. In this case, the receptor itself is an ion channel. So the transmitter hits the mitt, and bam, a gate opens immediately.
Like pushing a doorbell.
Exactly like a doorbell
Okay.
or a trapdoor. It happens in less than a millisecond. It lets specific ions pour through. If it's a cation channel, it's lined with negative charges, so it attracts and lets positive sodium ions in. This excites the neuron.
Any other type.
If it's an anion channel, its pores are sized just right to let negative chloride ions in while blocking the bigger sodium ions. This inhibits the neuron.
Okay, so that's for quick stuff. Moving my hand, hearing a sudden sound.
Correct. Direct, rapid electrical control. But there is a second type of receptor, and it's much more complex. The metabotropic receptor, which works through a second messenger system.
Why do we need a second, more complicated system?
Because those ion channels, the ionotropic ones, they close very quickly, within milliseconds. But think about your brain. You have memories that last for decades. You have moods that can last for hours or days. You need a mechanism that can cause prolonged structural changes in the cell.
You can't do that by just flashing a light switch on and off for a millisecond.
Exactly. You need to call in the renovation crew, and that's the second messenger.
So how does that work?
The most common one by far is the G protein complex.
G protein. Sounds complicated.
It's this little molecular machine that's attached to the inside of the receptor protein. When the neurotransmitter hits the receptor on the outside, it doesn't open a channel directly. Instead, it activates this G protein on the inside.
There's like a relay race.
It is. The G protein breaks loose. A piece of it, the alpha subunit, detaches and is now free to roam around inside the cell and, well, wreak havoc in a good way.
And what can this little free-roaming protein do?
It can do four main things that are incredibly powerful. One, it can wander over to an ion channel and open it and keep it open for a much longer time than an ionotropic receptor could.
Aye.
Two, it can activate enzymes inside the cell that change the cell's whole metabolism.
Okay.
Three, it can activate other chemical messengers inside the cell, starting a whole cascade of internal reactions. And four, and this is the absolute mind blower, it can activate gene transcription.
Whoa. It can talk to the DNA in the nucleus.
It can go right into the nucleus and cause specific genes to be transcribed into new proteins. It can literally tell the cell to manufacture new building materials. It can build new receptors or new dendritic spines to create new connections. This is the biological basis of long-term memory. When you learn something new, you are literally using G proteins to remodel the physical architecture of your brain cells.
That is wild. So a fleeting thought, if it's strong enough to trigger this system, isn't just electricity passing through. It leaves a physical footprint. It changes the building itself.
That's exactly right. This is why practice makes perfect isn't just a saying. You are physically rebuilding the circuit with new proteins to make it more efficient. And this connects back to what we were saying about personal identity.
How so?
Well, if your personality and your memories and your skills are just the specific arrangements of proteins in your neurons, and if G proteins are constantly changing those arrangements based on every new input you receive, then you are not a fixed object. You're a process. You're being chemically and physically rewritten every single day.
That's simultaneously terrifying and incredibly liberating.
It's just the reality of the nervous system.
Okay, we've been throwing around the words exciting and inhibiting a lot. But we need to do the math. Part 5 of our outline, the electrical math. Because at their core, neurons are basically tiny biological batteries, right?
They are. Every neuron maintains a resting membrane potential. In a typical spinal motor neuron, this is about negative 65 millivolts.
Why is it negative?
Because the inside of the cell is negatively charged compared to the fluid outside. There's an imbalance of ions.
And why negative 65 specifically? The notes mention that large peripheral nerves are lower, more like negative 70 or negative 90.
Negative 65 is a really interesting sweet spot. It's negative enough to be stable, but it's not too negative. It makes the neuron highly excitable. It's like a runner in the starting blocks, tensed and ready to go. If the resting potential were negative 90, it would be much harder to trigger an action potential. At negative 65, it's closer to the firing threshold.
Like a threshold is the tipping point.
Yes. For a neuron to fire an action potential, you typically need to raise the voltage from negative 65 millivolt up to about negative 45 millivolts. So you need a positive bump of about 20 millivolts.
So how do we get that bump?
That is excitation. We create an excitatory postsynaptic potential, or an EPSP.
Okay, we're getting into the alphabet soup now, EPSP.
It's all about concentration gradients. It's simple physics. The ion sodium, Na plus Fp, is highly concentrated in the fluid outside the cell. It desperately wants to get in. So when an excitatory neurotransmitter opens a sodium channel,
Sodium rushes in.
it floods in, bringing its positive charge with it. This influx of positive charge starts to neutralize some of that negative 65 inside the cell. The voltage begins to climb. Negative 60, negative 55, negative 50.
And if it climbs that full 20 millivolts to negative 45,
Boom! Threshold is reached, An action potential is triggered, the neuron fires, the gun goes off and sends a signal down the axon.
But obviously, we don't always want to fire. Sometimes we need to shut things down to say no.
That is inhibition, the inhibitory postsynaptic potential, or IPSP.
And how does that work? Do we just pump the sodium back out?
No, that would be way too slow and energy intensive. Instead, you do one of two things. The most common way is to open channels for the ion chloride, ClEl. Chloride is negatively charged and, like sodium, it's highly concentrated outside the cell. So if you open a chloride channel, that negative ion rushes in, making the inside of the cell even more negative.
So it pushes the voltage down from -65 to maybe 70 or -75.
Exactly. We call that hyperpolarization. You are actively pulling the membrane potential further away from the firing threshold. You are making it much harder for that neuron to fire, even if it's getting excitatory signals at the same time.
It's like turning up the noise-canceling headphones.
A perfect analogy. Or the other option is to open channels for potassium, K+. Now, potassium is positive, but it's highly concentrated inside the cell. So if you open the door for potassium, it rushes out. Losing a positive charge is mathematically the same as gaining a negative one. The result is the same, hyperpolarization.
And this dance, this constant battle between sodium rushing in for an EPSP and chloride rushing in for an IPSP is happening millions of times a second across the whole brain.
It is the fundamental algebra of thought. Every single decision you make, from what to have for breakfast to a major life choice, is just a summation of countless EPSPs and IPSPs in different neural circuits.
So let's talk about the chemicals themselves that are causing these potentials. We've been saying neurotransmitters generally, but the text divides them into two very distinct classes: the small molecule, rapidly acting transmitters, and the neuropeptides.
I like to think of them as the light infantry and the heavy artillery.
Okay, I like that. Let's start with the infantry, the small molecules.
These are your workhorses. We're talking about chemicals like acetylcholine, norepinephrine, dopamine, serotonin, GABA, glutamate, glycine. They have a few things in common. They are synthesized right there in the cytosol of the presynaptic terminal.
So they're manufactured locally, right on the front lines where they're needed.
Yes. And their vesicles are recycled. Take acetylcholine, for example. It gets released, it does its job on the receptor, and then it's immediately broken down by an enzyme in the gap. The choline part is sucked right back up into the terminal to be used to make a new molecule of acetylcholine. It's a very efficient, fast turnover system.
And what's their main function? What kind of jobs do they do?
They're responsible for most of the acute moment-to-moment signaling in the nervous system, transmitting sensory inputs, sending motor commands, move arm now, see light now.
Can we get into a few specifics? What does acetylcholine do?
Acetylcholine is, for the most part, excitatory. It's the transmitter used at the neuromuscular junction to make your muscles contract, for example.
And norepinephrine.
Norepinephrine is interesting. Many of the neurons that use it are located in the brainstem in a place called the locus coeruleus, but they send axons out to virtually every area of the brain. It seems to control overall activity and mood, like controlling wakefulness.
What about dopamine? That's a famous one.
Dopamine is crucial. It can be excitatory or inhibitory depending on the receptor, but one of its most famous roles is as an inhibitor in parts of the basal ganglia in the brain.
Wait, dopamine is inhibitory. I always thought of it as the pleasure molecule, something that's exciting.
It's far more complex than that. In the motor system, it plays a key role in inhibiting unwanted movements, allowing for smooth, controlled action. This is why in Parkinson's disease, where you lose dopamine-producing neurons, people develop tremors and rigidity. The inhibition is gone, and the motor circuits are running out of control.
That's fascinating. And then we have GABA and glycine.
The great inhibitors. GABA is considered the main inhibitory transmitter in the brain, and glycine in the spinal cord. They are the brakes of the nervous system. Without them, you'd have runaway excitation, which is essentially a seizure.
The notes also mention nitric oxide, which really stood out to me. It's a gas.
Yes, it's a total outlier. It's not made in advance and stored in vesicles like the others. It's synthesized instantly, on demand, in the terminal. And because it's a gas, it doesn't need a receptor on the surface. It just diffuses right through the cell membrane of the neighboring neuron and acts on metabolic machinery inside.
So it just goes through the walls.
Exactly. It's thought to be involved in things like long-term behavior and memory. It's a very different kind of messenger.
Okay, so that's the fast-acting infantry. Now let's talk about the heavy artillery, the neuropeptides.
These are a totally different class of molecule. They are much, much larger, and they aren't made in the terminal. They are synthesized on the ribosomes in the main cell body, the soma packaged by the Golgi apparatus, and then have to be transported slowly down the axon to the tip. It's a process called axonal streaming.
That sounds like a really long and inefficient commute.
It can be. The journey can be centimeters long, which in the cellular world is miles. It's a very slow process. And because of this, the vesicles they're in aren't recycled like the small molecule ones. Once they release their contents, the vesicle is gone, broken down.
So why on earth use them? Why go through all that trouble?
Because they are incredibly potent. The text says they can be 1,000 times more potent than small molecules. And more importantly, their effects are much more prolonged and durable.
How so?
While acetylcholine might open a channel for a millisecond, a neuropeptide might cause a calcium channel to stay closed for minutes or hours. Or it might change the metabolic machinery of the cell for days, or even alter the activation of specific genes for weeks. They cause much more profound, long-lasting changes.
So if I'm understanding this, small molecules are for saying move your hand right now, and neuropeptides are for establishing something like be in a bad mood all afternoon.
Roughly speaking, yes. They modulate the overall tone and long-term state of the system. And here's the kicker. A single neuron can release both. It can release a fast neurotransmitter from some vesicles to say hello, and with a different pattern of firing, release a neuropeptide from other vesicles to say, and remember this for a long time.
That just adds so much nuance. It's not just on or off. It's on, off, plus for how long, plus how strongly.
Which leads us perfectly into part 7, processing the signal, because, as we said, one single presynaptic terminal firing one little vesicle of transmitter only changes the postsynaptic voltage by maybe 0.5 to 1 millivolt.
And we need a 20 millivolt change to fire.
Right. So a single signal from a single input is, for all practical purposes, ignored. It does absolutely nothing on its own.
So how do we ever fire an action potential?
Summation. We have to add up the inputs. And there are two main ways the neuron does this: spatial summation and temporal summation.
Okay, break them down for me.
Spatial summation is basically voting by location. Imagine you have 40 different excitatory terminals firing at the same time, but on different physical parts of the neurons, dendrites, and soma. One adds 0.5 MV here, another adds 1 milliV over there. If enough of them arrive simultaneously, their individual electrical effects spread and literally stack up on top of each other at the soma to reach the threshold.
So it's strength in numbers from different places.
Exactly. Temporal summation is different. That's about voting over time. This is one single presynaptic terminal firing rapidly in succession. Pop, 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 pop.
Like it's piggybacking on its own signal.
Yes. The first release of transmitter opens some channels, and before the membrane potential can drop back down to rest, the second release hits, and the third. It builds on the shoulders of the previous signal, climbing higher and higher until it reaches threshold.
And what happens if we don't quite reach the threshold? Say we get a bunch of signals that add up to a plus 15 milliV change, so we get to negative 50 milliV, but we need to get to negative 45 milliV. What happens then?
That neuron is now in a state we call facilitated. It hasn't fired, but it is incredibly easy to set off. It's on the edge of its seat. A tiny little whisper of an additional excitatory signal that would normally do nothing will now be enough to tip it over the edge.
So this is crucial for things like attention or priming. If you're expecting an important phone call, your auditory neurons are probably facilitated.
Precisely. They are primed and ready to fire at the slightest sound that matches the pattern of a ringtone.
Now, the source material makes a really big point here about the special role of dendrites. It says dendrites rarely, if ever, fire action potentials themselves.
This is a key detail. The membrane of the dendrites has very few of those voltage-gated sodium channels that are necessary to generate an all-or-nothing action potential spike. They can't propagate a signal over long distances like the axon can.
So how does the signal from a synapse way out on a dendrite ever get to the soma where the decision to fire is made?
Through something called electrotonic conduction, which is a fancy way of saying it's just the passive flow of electrical current through the salty fluid inside the dendrite. But here's the catch. Dendrites are very long and thin, and their membranes are electrically leaky.
Leaky? That sounds like a pretty major design flaw.
It sounds bad, doesn't it? But it's actually a brilliant feature. Because the membrane is leaky, as that electrical signal travels down the long dendrite toward the soma, a lot of the current leaks out. The signal loses strength as it travels. This is a process called decrement.
Why on earth is losing signal a good thing?
Because it allows for analog mathematics. It allows an inhibitory signal that lands on the dendrite to physically cancel out an excitatory signal before it ever reaches the soma. If dendrites just fired all-or-nothing spikes like axons, you couldn't have that subtle subtraction and addition happening all along their length. Being leaky allows the neuron to weigh the evidence from thousands of inputs, giving more importance to synapses closer to the soma and less to those far away.
So it's the difference between a simple binary on-off switch and a sophisticated dimmer switch.
Precisely. It gives us nuance. It's part of what makes human thought so flexible and powerful, rather than rigidly robotic.
Let's move to the final section of our breakdown then. Special characteristics and, maybe more interestingly, vulnerabilities. Because this is a finely tuned electrical and chemical system, it can be hacked, or it can just break.
It's surprisingly fragile in some ways. Let's start with fatigue.
I'm certainly feeling that right now.
Synaptic fatigue is a real physiological phenomenon. If you stimulate an excitatory synapse rapidly and repeatedly, the rate of firing of the postsynaptic neuron is initially very high, but then it crashes after a few seconds or minutes.
Why does that happen?
It literally runs out of ammunition. It exhausts its store of neurotransmitter vesicles in the presynaptic terminal. The factory that makes the transmitter simply can't keep up with the demand.
Is that a bug in the system?
No, it's a safety breaker. It's a hugely important protective mechanism against excess neuronal activity. This is thought to be one of the primary mechanisms that helps to stop an epileptic seizure. The brain circuits literally tire themselves out to save the brain from damage.
Wow. Okay, what about pH? We know the body's obsessed with keeping its pH balance just right.
And the brain is exquisitely sensitive to it. Normally, our arterial blood is at a pH of 7.4. If you become alkalotic, meaning the pH goes up to 7.8 or 8.00, the excitability of neurons increases dramatically.
And that can cause seizures.
Yes. A very simple example. If you hyperventilate, you blow off a lot of carbon dioxide. Less CO2 in the blood means less carbonic acid, which means a higher, more alkaline pH. This is why forced hyperventilation can make you feel dizzy or lightheaded and can even trigger a seizure in people who are predisposed to them. Your neurons are firing way too easily.
And what about the opposite, acidosis?
A drop in pH down towards 7.0 causes a profound depression of neuronal activity. If it gets severe enough, it can lead to a coma. This is what happens in cases of severe diabetic acidosis, for example. The brain just shuts down. It essentially goes to sleep and won't wake up.
And oxygen.
The most critical resource of all. The brain has an incredibly high metabolic rate, but it stores almost no oxygen. It lives completely hand-to-mouth, totally dependent on continuous blood flow. An interruption of blood flow to the brain for just 3 to 7 seconds leads to unconsciousness.
3 to 7 seconds, that's it.
That's it. It crashes instantly without its fuel.
And finally, let's talk about drugs. Because we are constantly manipulating the system, intentionally or not.
We are. Take caffeine or theophylline in tea or theobromine in cocoa. What these substances do is increase the excitability of neurons, probably by reducing the threshold for excitation. They make the trigger a hair trigger.
That certainly explains the jitters I get after a double espresso.
I've
I've literally lowered my firing threshold.
You've made your neurons facilitated across the board. On the flip side, most anesthetics are thought to work by increasing the threshold for firing. They make the neuronal membrane harder to excite, often by changing the physical characteristics of the lipid membrane. They put the neuron into a deep sleep, so it ignores the signals of pain from the surgeon's knife.
And then there's something like strychnine, the classic poison from an Agatha Christie novel.
A really nasty one. Strychnine doesn't excite neurons directly. What it does is it inhibits the inhibitors. Specifically, it blocks the action of the inhibitory neurotransmitter glycine in the spinal cord.
So it cuts the brake lines on the system.
That's exactly what it does. And without the constant braking action of glycine, the excitatory signals run wild. The slightest sensory input can cause massive runaway muscle spasms and convulsions. It's a terrifying illustration of just how much braking is happening in your brain every single second just to keep you sitting here normally. Your brain is constantly holding itself back, and strychnine just releases that hold.
So let's try to synthesize all of this. We've gone on this incredible journey from a network of 100 billion neurons all the way down to a single tiny calcium ion slipping through a gate. We've looked at the spam filter, the walking subroutine in the spine, the leaky dendrites.
It really is a journey from the macroscopic structure down to the molecular machinery.
And what just completely blows my mind is that every single thought I'm having right now, every memory of my childhood that I can call up, every movement of my hand as I'm talking. It is just the sum of these little sodium and chloride ions rushing back and forth across membranes.
It is. We are a symphony of electrochemical gates opening and closing in an unfathomably complex pattern.
But here's where it gets really interesting for me, and maybe a little bit existential. We talked about those G proteins, the renovation crew.
Yes, the second messengers.
If our long-term memories and our very personality are built on these second messenger systems, physically changing the structure of proteins and even activating genes inside our neurons, and if those structures are constantly being remodeled, constantly being updated with new proteins based on our experiences, how permanent is who we are?
That is the profound question at the heart of neuroscience, isn't it? We like to think of ourselves as these static statues, but the biology shows us we're more like rivers. The water is always changing, the shape of the riverbed is slowly shifting, but we still call it the same river. Our self is a pattern of plasticity, a dynamic equilibrium maintained by G proteins and gene transcription. We are biologically designed to learn and to change. The you of today is chemically and structurally different from the you of yesterday.
And to think that the fact that our dendrites are leaky, that they lose signal, that this imperfection is actually what allows us to think broadly and with nuance.
Imperfection permits calculation. If every component were a perfect all-or-nothing digital switch, we'd be rigid robots. Being leaky and analog allows us to weigh probabilities, to integrate subtle cues, to ignore the spam so we can focus on the real signal.
Well, I think I'm going to be thinking about my leaky dendrites for the rest of the day.
As you should. They're doing a lot of work for you.
That just about wraps up our deep dive into the architecture of thought. It is truly humbling to realize that just to process the sound of our voices right now, your brain has just performed billions upon billions of calculations, filtered out the feeling of your socks, balanced your blood pH perfectly, and probably remodeled a few million proteins to help store this conversation as a memory.
It's the ultimate machine, running silently in the background.
Thanks for listening. Keep those synapses firing, and we'll catch you on the next deep dive.